Oh, can everybody hear me? Yes. Okay. Okay, so before I start with today's talk, I just wanted to announce next week's talk will be exploring the advantages of split pool combinatorial barcoding for scRNA-seq and sampling hematopoietic populations from adult zebrafish marrow using single-cell RNA-seq. So it will be next week, the 16th. You can visit biotech.wisc.edu for schedule and registration information for that. And before we get started with the talk, I just wanted to introduce the BioGarage. For those who don't know me, my name is Molly Nellen, and I'm a research specialist in the Advanced Genome Editing Lab here at the Biotechnology Center, and I'm also the manager of a facility called the BioGarage. The BioGarage is a shared lab space that provides affordable access to resources and services to aid in molecular biology research. These resources include a fully stocked bench space, as well as a cell culture room with a biosafety cabinet and CO2 incubator. We also have some basic workhorse services such as plasmid preps and mouse genotyping services. And the main service that I'd like to highlight for you today is our walk-up instrumentation service. So the BioGarage contains a number of instruments that can assist you in every step of your experimental process, from sample prep to QC to sample analysis. Firstly, we have a liquid chromatography system available for protein purification, as well as 2 QIAcube instruments that are able to automate more than 80 QIAGEN kits. Our Agilent bioanalyzer, qubit fluorometer, and nanodrop spectrophotometer allow you to assess quality and quantity of DNA, RNA, and protein samples. And lastly, we have several real-time PCR instruments, as well as a digital PCR instrument. And we are also planning to add an Illumina NextSeq 2000 in the coming weeks. And BioGarage staff provides training and consultation services for each instrument. And all of the instruments are available for you to sign up online and use whenever is convenient for you. So if you're interested in learning more about our services, you can reach out to me at BioGarage at biotech.wisc.edu or check out our website for more information. So now, moving on to our speaker. Susan Hetzel is a scientist with 12 years of experience as a researcher at the University of Minnesota in Minneapolis, where she studied the connection between biology and childhood psychosocial development. She has been a field application scientist at Thermo Fisher for 2 years, where she supports scientists with sample prep and qPCR and digital PCR applications. And she has been a huge help with our qPCR and digital PCR platforms in the BioGarage. So please give a warm welcome to Susan.
All right, so today the topic that we are going to explore is quality control in gene expression experiments. I know that's lovely and a nice title, but really my goal today is to give you the tools so that we can get you more publications and fewer reviewer comments back to you for additional information. Or if you do get reviewer comments for supporting information, which information should you have in your back pocket to provide to them? So let's start the presentation. So the talk today does assume some previous knowledge of qPCR. So I'm going to give you an introduction, but the full bit, I'm going to recommend you use our Education Connect website and go through this tutorial. I love recorded content such as this, because I don't know about you, if I run into something I don't understand, I can hit pause, Google, understand, links, references, come back to my recording and continue forward. So please take advantage of this. And there's many other resources on our website. But if you want any additional information, there's plenty of our Thermo folks here today, or through email, we can recommend other things besides this. So to sort of start this off, what is real-time PCR? After that, we're going to talk about quality and data patterns, and then move into talking about the core components of specificity, dynamic range, and ongoing controls. Just to let our online folks know, if you'd like to drop questions in the chat, we've got people monitoring that. So there will be able to convey that to me or respond in real time. So go ahead and take advantage of that. Or those of you here, I love questions and I love conversation. So just speak up. Okay, so what is real-time PCR? The essence of it is based on the traditional PCR application. And what kind of applications can we get out of this? A gene expression, that's why we're all here. But in addition to that, we used it en masse with our COVID response, so pathogen detection. There's single nucleotide polymorphisms. I personally have a lot of love for this application with my background. We have copy number variation, can be associated with very critical diagnoses. SCID, SMA, and spinal muscular atrophy in children is a really great example of a copy number assay. Mutation detection, DNA contamination, and the list goes on and on. But just to put a plug back in for Molly, if you have an interest in any of these, talk to me. You can use their equipment. They have top-of-the-line QuantStudio 7 Pro instruments that are very user-friendly. So please take advantage of that. What do you need if you're starting brand new to qPCR? Core components, you need a test. You need an assay. That's what we're looking at here. Whether it's Cyber or TACMAN, you need to target a gene of interest. Then we have master mix, and it is all those other components that you need to perform amplification that aren't part of the test. So we've got enzymes, NTPs, we've got maybe a passive reference dye, we've got salts and buffers. So all of those other components. The last two pieces are physical components that we need to actually run the test. We need consumables and plastics. Keeping our workspace clean and our plastics clean is always of importance when we do this. So keep that in mind when you're preparing to do this. Also make sure that the plastics you choose are compatible with the instrument that you're working on. If you have any questions about that, Molly would have a plastics compatibility chart, but if your plate doesn't fit properly, you won't get appropriate amplification. So please be aware of that. When we do qPCR testing, our first stopping point in the data journey is generating these CT values. CT values are unitless numbers we can gather and compare to each other to determine a difference in quantity. Ultimately, they don't track back to absolute numbers, but they are really wonderful tools to understanding what's going on in our experiment. Higher numbers mean lower quantities. Lower numbers mean higher quantities for CT. We determine a CT value at the threshold where our amplification curve crosses the threshold. A common question I get from new users who have done a lot of reading, but not necessarily as much experimentation, or maybe they have, is what is the difference between a CT and a CQ? The answer is somewhat surprising. In this instance, the way we're utilizing the data that I'm presenting here in this presentation, a CQ and a CT are the same thing. One is denoting CT, cycle at threshold. It's where our cycle crosses the threshold line. The other one is CQ, cycle at quantitation. So in essence, they're the same thing functionally in this kind of experiment. So this whole technology builds on PCR, and PCR is analyzable in early days, and anybody that's a little on the older side, gels. We used to run gels all the time. And it's easy to see from these gels, 1 versus 2 and 3 versus 4, that we have one of these two pairs has primer dimer formation or a satellite product, and the other one does not. Something to keep in mind when we're moving forward and talking about qPCR, some chemistries and technologies will report qPCR signal and others will not. Because in contrast, the gel we separate based on size. So they're different, we can separate them, and they're in two physical locations. This can't happen when everything's floating around in a single tube. So we need to think about that when we go forward and start designing our experiments, that everything lives in that one single tube. We're all here to talk about quality. So on the big picture, what is quality? It's a degree of excellence. That's a really big, glorious word. But in the lab, what does it mean? For scientific data, excellence and quality means accuracy, that whatever information you're putting out is accurate. If we're trying to quantify, which we are in gene expression for the most part, it's about precision. Precision and accuracy are two lovely friends. So if we don't have good quality, we typically are going to have a question about what we're actually looking at. But if we have high quality, we can move into the green and have a lot of confidence about what we're putting forward. To ensure that we have high quality, we typically, these are more industry related terms, but you've probably heard QA, QC. So quality assurance and quality control. It's about gathering evidence, whether you're doing it yourself or you're doing it as part of a larger lab, as part of, you're part of our team and you're on Thermo Fisher, and does the master mix work? Well, you've got to gather some data and figure out if it does or not. So a couple of things we're going to key in on today: assay specificity, sample quality, and then our targets. So when you're setting up and designing experiments and designing new systems, think bigger than the narrow approach that we're going for, because all of these things impact the overall quality of the system. Some of the applications I mentioned before, copy number variation and SNP genotyping, are a little bit easier to review for quality because the data is expected to come out in particular patterns. Copy number variation, this plot here, samples are either 1, 2, or 3 copies. If it comes out at 2.5, you know that sample's probably of lower quality. With our SNP genotyping, our assays, our responses are grouped into clusters. So you have something that falls outside of the cluster, you know that is aberrant data, or at least needs to be reviewed further. So those applications aside, we are here to talk about gene expression, which is where we take a gene, we biologically go through transcription and generate mRNA. So we're really, for the most part, trying to measure how much mRNA there is in a sample. One of the hurdles that we have with gene expression is we don't have hard and fast absolute numbers. Everything we're going to look at is based on RQ, or relative quantity. Everything is in comparison to something else. So we're not just relying on one test measurement. We're relying on multiple test measurements to generate the data we're actually going to move forward and publish. And even more complicated and more to the point is there isn't really an expected pattern for this kind of data. We have upregulation, downregulation, we have high increases, we can have log fold increases or decreases. and they don't necessarily fit into nice defined groups. So we're already up against a wall. So to combat some of these things, we want to talk about gene expression accuracy. And usually when I talk to people, the most common answer I get about what kind of minimum discrimination do we want to see between samples, it's twofold. If it's below that, you might be pushing things. In our product literature, we limit this to a twofold. And that's actually a really small difference. Remember, we have to get one relative quantification number, we have to do a minimum of 4 measurements. So getting a twofold difference out of this is actually a really small number and a good goal to have, but there really isn't much room for error. If we imagine we have 2 systems where we have low expression on the left and high expression on the right, or higher expression on the right, and we have error bars that are of a particular size, you can get statistically significant data out of the high expressor, but with the same kind of error, you are not going to have a statistically significant p-value in the low response assay. So the smaller we can make those error bars, the more significance we can add to your experiment and more publications that can go out. And that's really one of our goals as researchers. So can we have an expectation of the kind of data we think we're going to see in a gene expression experiment? Well, one of the common places that people look to for expectations in their gene expression experiments are translation experiments, protein expression. That's not entirely bad, but also not entirely accurate either. If we look at the expression profile of a particular gene in multiple samples, some from brain, some from liver, you can see that we have a different expression profile in each one of those tissues. It has the same genome, but different expression and potentially different translation. Also, we have a lot of things that go into that process of translation that aren't part of the transcription event. So if we have a control and treated state, we can imagine that both of these scenarios have the same 3 mRNA molecules. And in our control state, those 3 mRNAs create 3 proteins. In our treated state, we also have microRNAs. Those microRNAs are binding to our mRNA. So now in our treated state, we have just 1 molecule of protein that gets transcribed. None of those microRNAs, unless we go test for them, will be evident in our gene expression experiment. So it can be helpful to look to translation and protein expression to understand and maybe predict what's happening in our gene expression experiment, but it's not a guarantee. So now what? Well, let's talk about the transcripts that we're testing for. With multiple introns and exons in a gene, you can get different kinds of splicing events and you can get different genotypes that produce different transcripts. One test can test for multiple transcripts, and ultimately that's a good choice, having an assay that will test for as many transcripts as you want to look at. But ultimately in this experiment, we have all these different transcripts, but only the ones highlighted in green are actually detected by the assay we have. To make this even more complicated, some of the transcripts we're testing for don't create proteins. So again, our translation data is only partially informative. And even to a more direct point, if we pick a test that doesn't detect even our highest expressing transcript, we've missed all of that information. So again, quality in our gene expression experiments starts at the concept stage, not at the end. So once we've picked a test, what do we do with it to ensure that we have high quality data? The first step is assay validation. So we can imagine a system where we have our target molecules here represented in green and non-target molecules in red. Ultimately, if we got to pick, we would want to detect 100% of the target molecules we're interested in seeing and 0 of our non-target molecules, resulting in our result being as much green as possible. More towards what the real world represents, we have a situation where we have a smaller number of target molecules and a large number of non-target molecules. Each of those has a sensitivity, and even if we have a small amount by percent detection of our non-target molecules, it can absolutely overwhelm the resulting data. So gene expression demands specificity. If you don't have specificity, everything is sort of questionable. And this is what we see in most of our eukaryotic targets, our organisms, where most everything in that whole exosome is not what we're looking for. And we have a small amount that is what we're looking for. The trickiest scenario, and this does happen often in eukaryotic organisms, is this idea of a homolog. Homologs are products of evolutionary processes, and they are sequences that are highly similar to our target, but not exactly the same. So they may have similar functions and similar sequences and can confound the data we're generating because things we want to stick to just the target are sticking in other places. And the reality is there's lots and lots of homologs in any exosome that we're going to target for eukaryotic organisms. So what do we need to do? We have to test for this. We have to go in and challenge our assay and see how specific our assay actually is. A reminder of the 2 basic camps we have for chemistries. We have 5' nuclease chemistry, better known as TACMan. That is a brand name. It's our Kleenex to tissue as TACMan is to 5' nuclease assays. And we have cyber. Cyber is attractive on a cost scale, but remember, we've already established we need a highly specific test so that we can generate accurate results. And purely from a scientific perspective, if you're going to ask me, one scientist to another, which one of these should I pick to generate the best quality data, I am absolutely going to point you to a TACMan assay. So how are they different? So in a TACMan assay, we have 2 primers, just like our PCR reaction we started with. In addition to that, we have a sequence-specific probe. When that probe is bound in the reaction, so our red and green balls represent TAC, as those progress forward and extend off our primer, we reach the end of our TACMan probe. When the enzyme reaches that and can't progress through that, it comes off and cleaves that probe that's in front of it, and that gets released into solution, and the enzyme continues with the rest of that amplification. What happens is when we're going to the detection phase, the probe, when it's bound with the fluorophore on the 5' end and the quencher on the 3' end, those are locked together in three-dimensional space and interact energetically. So we shoot a photon in, the reporter and the quencher share that energy and it doesn't fluoresce. But after cleavage, when they're free in solution and we hit that with our light source, it excites the fluorophore, it releases energy, and we measure that. So some fundamental differences there about how we generate signal in a TaqMan assay. What happens if we have mismatches? We've already introduced this idea of homologs and how are we going to utilize our primers and probes, and what's it really going to look like when it starts to amplify. So here you can see we have a mismatch space in the middle of our TaqMan probe, and it binds, but it does not bind as strongly as if it had a perfect match. It has a significantly reduced TM, or melting temperature. We have this same sort of event with the primer, but it's still stuck. So the primer is stuck and it's going to start amplification. There's nothing that says if it's a weak but still bound interaction that it will be selected against. No, it's not. You have a free hydroxyl group on the end of that primer, and you will, if it is stuck, even in the wrong spot, you're going to start amplification. However, with the probe, what happens is that enzyme comes up to the end of that mismatch, and it can actually kick it off. So if it's a weaker interaction, you won't produce signal. So we've increased the specificity that goes along with what we're demanding from our gene expression experiment, such that only signal from a cleaved probe will produce fluorescence in our reaction. Again, we can have primer-dimer events. We saw that in our gel. Primers sticking to themselves, primers sticking to other primers, but they're far too short to allow for probe binding. And we would have the same scenario where we would most likely have mismatches and therefore no signal generation. Where do you go to find TaqMan assays? You can design them yourself. You can, absolutely. We can help you with synthesis, but we have 2.8 million, it's M with a million of those assays already in our inventory, ready to order from many, many different organisms. We recently, just within the last couple of weeks, changed our whole website. So I kind of want to highlight this really quick so you know what you're looking for when you go to the website. There's the website to search for assays. We can launch that assay search wizard. And I love how positive they are. Let's do it. Anyway, so I'm going to look for a gene expression assay. I'm going to walk through these steps. This one I decided was going to be human. When you start out and you just want to try something out, I'm definitely going to suggest a single tube assay. Sorry. And then we have, you can enter targets. This search bar will accept many different kinds of inputs. I chose a gene name because that's easy, but it can take cosmic IDs sometimes or RS numbers, depending on what you're searching. So go wild. Now, this is the review step. So what kind of information do you get out of this? And just to save you some pain, this isn't just a sales pitch for TaqMan assays. I love TaqMan assays. They changed how I did all my work in the lab and saved me a ton of time. But one of the things I did really, really love was using the website for information mining. There is a lot of information on this page that you can apply to your own work that isn't necessarily just about buying stuff. So what do we have here? We're going to sort to the top of the list something that we recommend. There's certain metrics behind recommending those, but the one thing I want you to see is this one has covers 16 different transcripts. And from the 5 we have on this list, it's nearly the highest. And remember our protein expression experiment, we want to cover as many of those transcripts as we can. Next, and probably really important to our academic friends, is seeing this note here that says 60 citations. That means this specific assay, not thermo assays in general, but that product right there has been published 60 times. If you're new to this field, you kind of go, everybody is publishing there and I want to get published, so maybe I should look there too. There's probably something important about the region or the gene, or maybe this one just works really well. But that's usually you're in for publications, is to kind of build on what other people are doing. So this is easily palatable. If we click on that, which it's actually a button, if we click on that, we open this. So you can see what other people are doing and you can link to those PubMed articles and see them in real time. So again, we're talking about mining for information, and it's all at our fingertips now. I really think this is just awesome. It gives you a BioStars rating. So this is 93 out of 100. That's really good. That means people are publishing with this information, which we could see from the number of articles. In addition to that, if we click on the details tab, we can get a lot of information about the gene itself and also the different transcripts. So again, this is not a static website for ordering. This is really a repository of information ready for you to look at. I didn't include it in a slide, but we also have disease panels where you can start with a big idea of a disease and start drilling into different pathways to generate information, or maybe look at links that you can't see in other networks. So take advantage of all of that. I think it's great. Back to our red and green lines. Okay, so ideally, we want to have as much sensitivity towards our target with the least amount of non-target molecules, detected as possible. And with our TACMan chemistry, we can do that reliably. And believe it or not, we have an inventory of 2.8 million assays. They're wet bench tested. I did not believe that the first time I heard it. When I joined Thermo, I'm like, yeah, that's a great product line. So I tested it out. I grabbed a couple of assays at random from the website and I requested the QC data and they gave it to me. Yeah. Somebody actually sits down and sees how specific is this assay and does it actually work? To the point where we have a TACMan assay guarantee. If it doesn't perform, with some caveats, obviously we need to discuss the specifics, but within reason, we'll replace it or refund it. So that is really powerful to say it will work and it will be not only will it amplify your target, but it will do it specifically. So what is the actual requirement? If we compare a target to a homolog, our scientists sat down and said, how much difference do we need to have to guarantee performance? And they decided a delta CT of 15 of target versus homolog would be a good data point, which what it comes down to is a 30-fold difference in value. Way overkill. but that's what we do. That's what we offer. And somebody really tests this. So when we look at product numbers, what are we actually looking at? First two letters are your organism, HS, Homo sapiens. We have a assay number, and then the suffix is what is important. M1 is always going to be your first choice. That means it's over an exon-exon junction. If we can't meet our 30,000-fold guarantee, we give it an H. So if you see an H and you can pick a different assay, please do. So we still have a few of these, and it's just indicating that it's important and we have it, but we know it's going to have reduced specificity. And here's the other suffixes to look at. But again, your first stop, gene expression, if you can, M1. So that is really how we look at validating assay specificity. Unfortunately, when we look at cyber, we can't challenge the assay in the same kind of way. But what can we do? So with cyber, remember, anything and everything that is double-stranded, whether it's specific or not, is going to give you signal. Target, non-target, and primer dimers. So what can we do to look at assay specificity? If you saw this amplification curve, you would say, it's kind of hard to see in this light. It's in green. I would have picked a different color, but there it is. If I saw that amplification curve, I would have given it a thumbs up. That looks good. But it was run on a gel, and we can definitely see there's more than one product there. It's a little blurry, but there's actually 3 bands there. So what can we do? We can run everything out on a gel. We can do sequencing, and we can do melt curves. So in a gel, what are you looking for? You want one tight band with an approximate size of what you were looking for and nothing else. That's great. That is one piece of evidence that says you did it right, but it doesn't guarantee you did it right. Because remember, we have all those homologs, and they're as much as 90% similar, depending on what kind of homolog it is, which means you could have a homolog co-migrating with your target here, and you would never know it. So it's a good piece of information and adds to the story, but it's not definitive. You can run sequencing. Also great, real expensive. And potentially, depending on how many targets you have, you could get different sequencing results. But again, it's good to have it. I do recommend it at once at a minimum, but it's, again, a piece of information with an indication, but not definitive. Melt curves. Again, great information. What can we learn from melt curves? Lots of people do it. We start at a low temperature and we ramp up to a high temperature and collect fluorescence along the way. Remember, cyber is a double-stranded binding dye. So at low temperatures, we're going to have a lot of signal. And then as we slowly increase, we're going to peel those double strands apart and decrease that fluorescence until we get to a critical point along the way where we have this magical big event and everything falls apart. We call that our melting temperature, or TM. It's difficult for human eyes to visualize this. So if we take the first derivative of it, we can get a nice sharp peak. So when we look at this, the hallmark of melt curves for good data, we want a single, it's the 4 S's, single, smooth, sharp, symmetrical peak. Those are all indicators that we have a single product. But what can it prove and what does it not prove? Unfortunately, when you add cyber dye to a reaction, it changes melting temperature. So we can calculate what the TM should be, but when we actually go measure it with cyber, it's going to be shifted a little bit. So there's a little bit of ambiguity built into it right there. What can we also say is we have one length peak, but that doesn't guarantee it's the target we're looking for. Remember, our homologs are going to be quite similar, and they could have a very similar or identical melt temperature. What we can say is if we have multiple peaks, that's bad. So multiple peaks, meaning you have 3 distinct products. If we have a scenario where peaks from non-target molecules are shifted, that can be good evidence. But if they're in a mixture, you're going to ever so slowly minimize the difference. So if you'd have seen this melt curve alone, you probably would have thought it was okay. But in some of these samples, you can actually see this double hump, meaning we actually have 2 products inside this one larger peak. So it can be deceptive. And again, this is that gel from before. We ran it in a melt curve, and it gives us what really kind of looks like a single peak. It's not bad. So again, melt curves are great. It's great for QC, but it's not necessarily definitive. Blast. Again, when you design things, you want to start at the beginning and think about all the caveats. And one of them is non-specific binding. We can blast these sequences, but make sure you're including partial homology in that blast search. That again is going to help you, but not always be definitive. So please always blast them and look for that partial homology, not exact matches. Okay, it is a selection within the page. So make sure you use that. All right, so that's what we can do with our cyber data versus our TACMAN data for validation. Next, let's talk about dynamic. Yes, Alex.
Do you want to put a question?
Sure.
They're talking to this mic. Okay, so the question in the chat is, once we have designed primers, we can figure out whether primers can bind to homologs. Is that correct?
With TACMAN assays, if they're purchased from Thermo Fisher, we would have designed against that and done the wet bench testing for you. If you're doing it for yourself with cyber, unfortunately, there's no guarantee that you can definitively say, yes, I am. We have different metrics that indicate that you are, but there is no absolute, unfortunately.
Follow to that is why? Why is there no absolute?
I would turn that back around and challenge that with an additional question. Why? If you are a researcher, how would you do that? We're sort of limited with the tools that we have. We're trying to sequence a mixture of a population. You could try to run it for NGS. That seems like a very expensive solution to a problem that you could avoid by picking a different kind of chemistry. So could you conceptually? Yes. Would you effectively? No. With the testing that, unless I've missed something, my understanding is that's not possible with cyber. Okay. That is a very good question. I like that. So our second pillar, we'll move on quickly from here. Dynamics range testing. You need to make sure when you do your gene expression testing that you're in a range where everything works appropriately. And really, you've got this sliding scale where you want to keep all of your data acquisition inside that dynamic range. At the high end, you can have too much sample. You can overload your reaction with target molecules. You'll get reduced response and it won't look right. On the lower end, you can have inadequate sensitivity. So you have unreliable data because there isn't enough sample in your measurement. So that's where things fall apart. That's really high CTs over there, high numbers, 39, 40. That's on that end. So how do we do this? So I actually recommend testing both cDNA input in a serial dilution to test for gene expression dynamic range, And there's one that everybody forgets about. And that's the one I really want to highlight right now. And that's doing a serial dilution of your RNA into your reverse transcription reaction. Gene expression is great, but if we're doing this in 2 steps, we're having 2 different enzymes and they can have 2 separate problems. And we've got to test that. You don't have to test every single sample, but you've got to test a range of samples to make sure you're accurately copying your RNA to cDNA. We have 2 different kinds of master mixes on our website. We can do this in a 2-step reaction where we do reverse transcription, we generate a cDNA library, then we come back and we do our gene expression testing from our cDNA, or we can do it in a 1-step. And that is 1 master mix that has 2 enzymes in it. You put RNA in the beginning and you get gene expression results out at the end of that experiment. Those are both available on the website. But what happens if we overload our RNA standard curve? So the idea is we've tested our assay in a serial dilution and we know what ranges it works in. Likewise, we titrated RNA into our reverse transcription reaction and then take that result and measure our expression. And we can see at what point are we overloading our reverse transcription reaction. And in this example, over here at our higher CT values, we actually see this hook up. And at some point, we have added so much stuff to our reaction that the enzymes aren't effectively copying everything that we want them to. And that is the upper limit for dynamic range of your reverse transcription reaction. Even when I was in the lab, I didn't do this. I was bad, but maybe that explains why I had some failed projects. Don't be me. So this is critical. You don't have to do it every single time, but you do need to do it. Also, the quality and sample preparation of those RNA samples can matter. This information came in from a customer. We were troubleshooting this as a team. This is what their data looked like. They titrated in their RNA. They should have had slopes here of about 3.3. And every single time, every different sample they put in, they had a different slope. Bad news. If you were running your gene expression experiments after this reverse transcription reaction, you would have no idea what your results were. You were basically generating random numbers rather than actual gene expression information. So we gave them a different product to try out. They did the same, took the same samples, put them into a different reverse transcription reaction. And now that serial dilution gives us nice straight lines with wonderful slopes. Yay, high capacity wins. It's still my favorite because I'm a dork and I have favorites. All right, so everybody likes to skip that step. I was guilty of it too. Be better than me. So that's dynamic range testing. Please do both cDNA and RNA. The last one is ongoing controls. This is something you do every time or regularly when you run experiments. Not every time, but regularly. So ideally, we would want to have both positive and negative controls. Multiple different kinds of negative controls. You can have no sample controls, something that's just water. You can have a negative control. That would be something if we were doing pathogen testing and we had a sample collected from somebody that we knew did not have the virus we're looking for. Those are negative controls. But again, the one I'm guilty of not doing as often as I should have is including this no reverse transcription control. We need to make sure that our reverse transcription reaction is appropriate. All right. I'm going to skip that. Positive controls. Wouldn't it be great if we had positive controls for gene expression experiments? To my knowledge, these don't exist. It would be great. Wouldn't it be great? But if you have something you can come back to and get a general idea of what the reaction should provide to you for a result, that would be great. Now, on the data side of things, I'm not going to spend a lot of time here because we're almost up on time, but just a reminder of where this information comes from. And I think it's funny to look back at this date. It's December 11th, 1997. That was 26 years ago. But we still use it because it's so important. So this is the idea when we look at gene expression, we're coming back again and looking at our delta CT, 2 delta CT, relative expression, lots of names for the same thing. So if we have this idealized data set, or maybe it's a real data set, I don't even know. I don't remember anymore. We need to have at least 4 components, 4 individual test points to ever generate a single RQ value. You need to have a sample for reference, so untreated or normal. In this, we are going to use brain. You also need to have something you're comparing it to, a treated sample, time course, a tumor, something like that. You also need to have 2 tests. You need to have a normalizer gene, endogenous control gene. Those are ideally not going to change at all under our experimental parameters. They're a constant that we're going to compare to. And you need to have something you're actually testing, the gene you're actually testing. So we ran this experiment and this is the data we generated. What do we do with it? Remember, we're talking about 2 delta CT. So the first step is to compare the CT values of our target versus our CT values of our normalizer. That's the first delta. It's a straight up subtraction. That's our first delta CT. Second delta is comparing the samples to each other. Remember, we're going to use brain as our reference. So we take the brain values and subtract the delta for our liver and our lung samples. We now have 2 delta CT. Completely meaningless numbers to my brain. How do we get meaningful numbers? We convert the 2 delta CT into RQ. That's relative quantity. So now, by definition, an RQ of 1 means no change. So the RQ of 2.6 for our lung sample means the expression for that lung sample for this target is 2.6 times higher than the brain sample. A fractional value for RQ means a reduced expression level. So 0.5 would mean we had half the amount of expression in that other sample as the brain sample. So the summary to what we talked about today, we require for gene expression high specificity. We need to have a chemistry that matches that, and we need to actually do the testing to make sure the data we gather is what we think it is and means what we think it means. So how do we do that? We measure assay specificity as much as possible. If it's TACBAN, we can interrogate that directly. If you buy it, we already did it for you. If it's cyber, you're going to have to gather those individual pieces of data and put them together into a bigger picture. You need to go in and test your dynamic range of both cDNA and reverse transcription. So an RNA standard curve, and choose appropriate controls. No template controls, knowns if you have them, negative controls, no RT controls. They're all very appropriate. So if you want, yeah.
Okay, a couple more questions. We'll start at the top. There's 3. Okay, it says, how do you get around primer efficiency in AT-rich genomes? Questions from...
Well, if you can't get around, so obviously you'd want to first tap into your amplification parameters and make sure you're looking at those and being critical about what you're actually making. But if you can't get around that, in our software, you can actually go in. If you do that cDNA Sander curve, you can actually go in and recalculate and add the efficiency number that's appropriate for the test you're actually performing. So if you know you're not getting twofold efficiency, twofold growth, you can adjust that and make your numbers reflect what's actually happening.
So you developed that
Yeah.
specifically. Okay. All right. I think that answers that one. The next question, what are your thoughts about using a modified delta-delta CT method that incorporates primer efficiency in the calculation?
I like it. I just suggested it. Yeah, I think it's great. If you know your assay isn't efficient and you're unwilling to redesign it to make it better, then yes, you really should implement a modified calculation. Absolutely.
Okay, last one is, my question is, if we duplicate in a plate, I'm assuming technical replication, how much standard deviation is acceptable?
That is a good one. And again, I have to put it back on you. What p-value do you want for your publication? I mean, if you have a really high expressing target, you don't need to have a teeny tiny error bar on it. It can be big. It's still statistically significant. If you're looking for that low end fold change, that twofold change, you're going to be much less tolerant. So unfortunately, the answer is it depends.
Right. And potentially qPCR isn't your answer. If you're looking for a very low end fold change, you might need to do something else.
Right.
That's a little more sensitive.
Maybe like digital.
Maybe digital.
Maybe digital.
Okay, that's all we have.
Awesome. Go ahead.
So hi, thank you for the very interesting talk. I'm Tanya. I am a graduate student in the Amman lab. So I have a couple of questions. So the first question is, what is the range of CT value you would recommend the expression profile being within? Also, the second question that you talked about, the endogenous controls. How do we select the best endogenous control for a particular set of experiments? Because personally, I have seen that using one kind of endogenous control can skew my data in a particular direction and the other can skew it in the other direction. So would you in that case recommend using more than one endogenous control? And if yes, then do you have any like mixture or high throughput type of endogenous control where using which I don't need to waste a lot of wells in my plate and just have one well, have all my endogenous control for a particular group of samples? Because if I'm trying to do high throughput running, I don't want to waste a lot of my well space with the endogenous control.
Right. Sure. There were three questions. What is a CT range that's acceptable for a gene expression experiment? The second one was about endogenous controls and how do you pick it? Third, do we have multiple endogenous control pools, basically? We'll start at the top. Those are amazing questions. You're thinking critically about what's going on, and I love to hear that. So first of all, CT range. I'm going to point you back to dynamic range testing. The instrument has 9 logs of dynamic range. So what you need to do, I typically don't recommend looking at anything that has a CT below 10, 8 to 10. It just makes it difficult for the instrument to accurately identify a CT value. So that's kind of your high end, and then your low end, anything above, I would say if you're being critical, anything over 36, maybe 37 is in the range. If we go through and do the calculations and say I have 1 molecule or even 10 molecules and I have a twofold amplification, when am I really going to see it? And that ends up in that 36 plus range. So I would stick in that range, but ultimately it's going to be limited by your dynamic range testing. So go there. Next question about picking an endogenous control. That is an excellent question. And there's a lot of debate in some of these areas that leads into how to answer your third question. But there is no substitute to testing several endogenous controls and seeing which one is the most functional.
Yeah, so with that endogenous control, like there are a few tested endogenous controls that work, but we kind of see sometimes that compared to one endogenous control, I can see an upward trend, compared to the other, you can see an upward trend, which is.
Yeah, it's concerning. And that's where I would maybe start thinking about a third, because I'm curious if you see a trend in one direction with one reference and a trend in an entirely different direction with another, how do we know which one is right? How do you? So I would go ahead and throw a third or a fourth. We sell endogenous control plates for this very purpose, because if you're walking into a new system, how do you know which is the right endogenous control to pick? So we have these plates that are already made up with our top 12 or so endogenous controls available. You can run on a few samples and see which ones are the most stable, which one works for your experiments, because there's no substitute for that. 18S might be perfect for one experimenter under particular conditions, but that's not necessarily what you're doing. It might not be appropriate for your experiment. There's no slam dunk, essentially. So that leads into your third question about pooling. So we do have these plates, but we provide them to you discreetly. They're individually. We don't advocate for pooling, because ultimately your pool might be 3 good ones and 1 bad one. You've now summarized the data with all 4 of those.
What I meant is, like, since the stackment rows could be tagged with different rows, could we have different colors of each of them without having the fear of bleeding one color into other, like just discrete colors in one group?
We're really good at that. It's very easy to duplex. A lot of people do it without even experimenting with it. They just throw them in and they're done. And that's 2 colors. You could potentially do 3 if you wanted to. Yeah. And then decide when you're done. You have to do validation and work up to it. You need to run that dynamic range testing and run a standard curve for all of the singlets, then all of the 2-way reactions, and then all of the 3-way reactions, and build up until you can be certain that your reaction efficiency, your growth of product is not changing by adding different tests together. So that's the correct way to go about it. Do your testing. Okay. Duplex is very common. Triplex happens, but is rarer. Quadruplex, we only have done it regularly with pathogen detection. So that is not quantitative at all. So I'm, especially in the research world, it's a very time, labor, and cost intensive to do anything more than 3. Even 3 can be quite daunting. Yeah, if
we can get you set up.
Yeah, we on our website, any of those. So if we go back to, we have a sale on TACMAN assays. Talk to Alex.
Yeah, and I have this for people who are here as well, like a birthday flyer you can take, but it does include some of the big assays which can help with your specific thing. If you want to try duplexes, that type of stuff.
Yeah. So when we go, I'll just come back here so we can see it really quick. But right here where it says related products, you can click on that and it will recommend you some endogenous controls that might work for your test. And when you order those, you can come in under this tab here where it says FAM MGB, and you can select a VIC, or VIC is web orderable. Anything else will need to go through specialty oligos to get prepared for the other colors. But FAM VIC, you're off to the races. Order that when you leave here today. At a discount.
Yeah.
60%.
One more question.
Yep.
Okay, so I thought I answered this and then maybe I was mistaken, or I don't know. It was on your slide exactly. But the question was, how can we do protein quantifying by qPCR? My response was, you can't. There's no DNA in protein in the short version. And then Cody is mentioning that in one of your earlier slides, it mentioned that and protein stability.
Right. So
did I miss something?
Yeah. So we do have a kit we call protein thermal shift that a lot of people like to use to look at protein folding. It's a special dye. It's not a DNA application. It's not often done here in the central part of the US, really more common on the coast, Boston and Tech in Southern California. They do it a lot there. But essentially you look at the temperature, change the temperature over time, and watch that fluorescence change as the protein unfolds and refolds. So
They're not quantitative of how much protein is there.
yeah, is protein quantification on there? I'll have to go look. Potentially. Not a common application, that's for sure. It does say protein quantification. I'll have to talk to John Pfeiffer about that. He'll know. He's our resident guru. He actually predates qPCR. Can you believe it? Yeah.
I have a question. When did you choose one step over two step to qPCR?
Right. Great question. If you want to build a cDNA library, you're not going to choose two-step, or you're going to choose two-step, not one step. If two-step is less expensive than a one-step, but one step is going to account for all of the pipetting steps. So you're going to have less variability in your end result. So if you can afford and have a system that responds well to the one-step chemistry, I would recommend it. It's fewer plates, it's fewer pipetting steps, sample to answer. We even have a cells to CT where you go right from whole cells and you get a gene expression result at the end. So
Okay,
we have all of those. Yeah.
Thank you. Great seminar. Do you remember the Mikey guidelines? You did a great summary representation of all the things you should look at. Have those been updated at all? Do you know?
Not substantially.
Do
There's
people
been
use
minor.
them?
I really don't think they're terribly appropriate. And to be honest, a lot of reviewers that I've found don't understand what they are to begin with. So, I mean, to be perfectly honest, I mean, that's kind of cutting, but that's the reality. So Mikey guidelines are just that, they're guidelines. They're not exclusive because Mikey isn't the one that's defining whether you get published or not. So that's a great question that I'm unsure about the best way to answer. But I think they've been updated in the last, I think 3 or 4 years, but not since then. And even then, I don't think they were substantially altered. Yeah.
Just one last question. Have you guys checked for the bleeding between FAM and VIC?
Oh, yes, that's actually, the question is, have we checked for bleeding between FAM and VIC? If we look at a duplex assay and we have them both together, that's an excellent question. And actually we expect that, calculate for it, and then take it out of your data before we ever present it back to you. So that's part of the calibration that we do and why we recommend doing calibration on a regular basis, because we want you to have the most accurate data for what you're looking at, and that's part of the process. Yeah.
Thanks, Susan.
All right. Thanks, everybody.